Alright, it's Monday. I'm going to the lab to quickly inoculate some colonies for tomorrow, and after that, around at 5:30 p.m, we have a team building. Happy Tuesday, it's a little later, 10 a.m, because I went to the doctor to take my blood to analyze it for another video. Stay tuned. But now another day in the lab, a whole day. Let's go.
The grown colonies will be used for two experiments. For the first one, I just need to grow them further in a larger bottle. For the second experiment, I need to do a plasmid extraction. Therefore, cells are first separated from the media. Next, the cells are lysed to release the plasmids in the solution. Then, the white cell waste is separated from the yellow solution containing the plasmids by centrifugation.
Alright, it's 1 p.m, I'm getting some food. Meanwhile, behind the scenes, I did another Gibson assembly. I measured the DNA concentration of the plasmids from the plasmid extraction, and I made some glycerol stocks of those same colonies. Yeah.
Here I perform a third additional experiment in which I insert a newly created plasmid obtained after a Gibson assembly in some E. coli cells through a heat shock. Here, sock media is added in which the cells will be incubated for an hour. This way they can recover from the heat shock. After this, I plated the bacteria out on some petri dishes I had already prepared. Here they will grow on and hopefully create colonies. I use this Pasteur pipette sterilized by flame and ethanol to spread a droplet of bacteria over the plate.
I did a lot of stuff today. It's probably hard to follow because I'm doing a lot of experiments simultaneously. I did the plasmid extraction for one, then the Gibson assembly and a heat shock after that for something else. I'm also growing some other bacteria for the Western blot and the AKTA purification. Alright, the final thing I did is adding IPTG to induce those 3 bottles there. And now I'm just going to work on a task of biochemical and molecular nutrition. Wednesday, working on projects. Happy Thursday!
Here I'm filter sterilizing a solution I need further on. I needed this filtered solution for this device. I'm going to perform an AKTA purification of the protein solution that my bacteria produced in the 3 larger bottles from Tuesday. The goal is to separate my protein of interest from all other proteins with an Affinity chromatography column. I have a very interesting separate video about this principle, link in description. But to summarize, the goal is that we're ending up with one solution containing only the protein I'm researching, and one solution containing all other proteins the cells produced. To do this, the machine first needs to be prepared and installed, which you see us doing here. The protein solution will follow this path through the tubes, going over the column, and different fractions will be collected in these tubes. The AKTA was running as it should. I performed the AKTA purification for 3 different proteins of interest. The first one is done, so here I'm switching between solutions. After the AKTA purification, the fractions are loaded on a protein gel to separate them based on molecular weight.
Alright, the SDS-PAGE shell is running. And then after this I'm going to stain one of the 2 and the other one is for a Western blot.
When the gel was finished running, I used it for a Western blot. Here the proteins are transferred to a membrane. In the following steps, only my protein of interest will be visualized by binding with specific antibodies. Alright, 6 p.m. the Western blot membrane is incubating in milk at the moment. Now I'm going to change it to the first antibody solution, which will incubate until tomorrow morning. This day I also performed another experiment in between. Remember the heat shock I performed on Tuesday? Well, the bacteria grew on the plate, and here I prepared the colony PCR for 8 different colonies to check if they contain the plasmid or not. Alright, while the AKTA is running, I'm going to go further with the PCR experiment. PCR is done. I prepared my DNA samples to load them on this gel. Let's load it on gel, shall we? And the result of the gel. Awesome. Late night workout. The following morning, the Western blot membrane was incubated in a second antibody solution. Finally, a coloring solution is added which will interact with an enzyme present on the secondary antibody and thus visualize my protein of interest. Meanwhile, it's almost 1 p.m. I also did another colony PCR of 15 additional colonies, as the same one as yesterday. Yesterday they were all negative for multiple colony site 2, so hopefully here one colony will be positive, but I have to hurry because I have to go to another campus in a few moments for a practical course. And just as always, finishing off the week with some editing so you guys can watch these videos. Subscribe!